Proteins can serve as molecular motors, and one of the best examples are the motors that power ATP synthase. Remind you here of the structure of amino acids, NCC, 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 with the R group coming off the middle carbon. In the case of glutamic acid, GLU, or E is its single letter abbreviation, it's an acid, a hydrogen H+, tends to leave under physiological conditions, and that leaves behind an oxygen that is negatively charged. We can refer to it as GLU-, GLU-. Arginine, ARG for 3 letters, R, is a basic amino acid. It tends to attract a proton H+, so that it's removing protons from water. Glutamic acid is an acid because it loses H+, arginine gains. Those are the 2 most important amino acids in this example I'm going to give. Here's the motor part of ATP synthase. It's embedded in the membrane, and the key parts are a row of glutamic acids. Remember, those are E's, and I'm drawing the minuses underneath. They're not underlines, they're minus charge. Here's a key arginine that's not on this cylinder. It's on another part of the protein that's on the side. Each of these E minuses on the edge have an H+ associated. The middle one near the arginine does not. On each side of the arginine, there's a proton half channel, and one side goes down to the matrix, and the other side goes up to the intermembrane space. There's 2 different parts to the quaternary structure of this protein. One is the cylinder, and the other is the proton half channel, the arginine, and the other proton half channel. If we looked at it from above, we'd see the cylinder embedded in membrane, and then a different protein that's attached to the cylinder holds the proton half channel that brings protons in and the proton half channel through which the protons leave. The positively charged arginine is also here. The E minuses are back in the cylinder. So protons are going in, wind up bound to an E minus, they rotate around, they come back out through this half channel to the other side of the membrane. The intermembrane space has a pH of 7, the matrix pH 8. And that means there are 10 times as many protons in the intermembrane space. The matrix is negatively charged, minus 150 millivolts, and that means there are unbalanced charges, chlorides, proteins, other negative charges unbalanced on the inside. The combination of those 2 mean that 1,000 times more likely for a hydrogen to enter a half channel from the intermembrane space. And when that happens, that attracts that glutamic acid. That turns this whole cylinder. The arginine stays in place, but it turns the cylinder. So the cylinder rotates because that glutamic acid got attracted to a proton. Next, glutamic acid moves a proton closer to arginine, and the arginine repels it. And so the H+ moves through the other half channel into the matrix. Once there, it's quickly attracted to one of these unbalanced negative charges. Now we have to move each of these glutamic acids over one. The arginine R stays in place, but the whole cylinder rotates. I letter these A, B, C. I'm going to re-letter these because I've moved the A, B, C, D over. We've moved all of the glutamic acids, one glutamic acid over by moving a proton in from the intermembrane space and out into the matrix. That rotates a shaft. That's what does the work of adding a phosphate to an ADP, but that's another video. Here's a quicker version of the video showing the cylinder inside the membranes, looking at it from the side. Glutamates, negatively charged. The 2 protons. Arginine, positively charged, and the 2 proton or H+ half channels. Matrix at the bottom, the intermembrane space at the top. pH is 7 in the intermembrane space, pH 8 in the bottom, and minus 150 millivolts on the matrix. So that means there are far more protons and free to move. The protons in the matrix are mostly interacting with the excess negative charge. So the result is 1,000 times more likely for a proton to enter than leave. When it does, that attracts a glutamic acid and turns our cylinder relative to the arginine. The turning of the cylinder brings a new hydrogen from that next glutamate over on the left near arginine, but arginine is repelling it, and so the H plus gets pushed out through the proton half channel. Once it's pushed out, that proton quickly associates with all the negative charge on the inside. That rotating of the shaft is what's going to do the work of adding phosphate to ADP to make ATP. Here's an image of what the cylinder and the half channels actually look like. All the gray is the cylinder. Each red area is one of the glutamic acids, GLU minus or E minus abbreviation. The green are the two half channels. The arginine is in blue in the middle of the green. I drew the half channel straight up and down, but actually they're sloped. The sloping doesn't really matter, so you can draw them either way. Here's a space-filling model of the entire ATP synthase. I've put in the location of the bilipid membrane. The part I have diagrammed in this video is in the box up at the top. The gray, again, is the cylinder that turns. The green are the half channels. I didn't sketch out the blue or the red portion. Here is this portion of the protein. It's a ribbon diagram. You're seeing a whole series of alpha helices of amino acids. Of the gray portion, the half channels are made up of about 4 to 6 helices. I've shown the direction of rotation. In red, the glutamic acids. Blue, the arginine plus. That's on the green, that's part of the protein that makes up the half channels. The gray rotates in the membrane, but the green stays still.